Alright, let's go. Number 10. The Barreleye fish's transparent skull. In 1939, ichthyologists working off the California coast pulled up a strange, flat-bodied fish with tubular eyes and gave it a name, Macropinna microstoma. Then filed it away as a curiosity of deep-sea taxonomy. For 65 years, nobody knew how its eyes actually worked, because every specimen hauled to the surface arrived with its transparent head collapsed into a ruined pink smear. Standard vertebrate anatomy says eyes are fixed in the skull, aimed forward or sideways, unable to independently rotate while an intact protective dome watches over them from above. In 2004, marine biologists Bruce Robeson and Kim Reisenbichler at the Monterey Bay Aquarium Research Institute sent a remotely operated vehicle 900 meters down and filmed a living barreleye for the first time. And what they saw made no anatomical sense. The fish's entire head was a fluid-filled, transparent dome, and inside it, two barrel-shaped eyes rotated independently, swiveling upward through the skull to track prey directly above, then pivoting forward again to align with the mouth. Robeson and Reisenbichler published the finding in Copia in 2009, describing a skull architecture with no equivalent anywhere else in vertebrate life. Early theories proposed the transparent shield was a fluke of pressure adaptation, a simple lens cover. That failed once researchers realized the dome is load-bearing and structurally essential, not incidental. Others proposed the rotating eyes were vestigial. That failed too, once footage showed the fish using them to steal food directly from the stinging tentacles of Siphonophores without triggering a single nematocyst. The visual system evolved to solve a problem so specific, hunting through another predator's weapon, that nothing comparable exists on the entire planet. Somewhere below the sunlit water you have swum in, a skull houses a brain watching the world through its own bones. What else is evolution built down there that simply never surfaces intact enough for us to see it working? 900 meters down, the pressure problem only gets worse from here. Number 9. The deep-sea brine pools. In the early 2010s, NOAA's Okeanos Explorer program was mapping unremarkable stretches of the Gulf of Mexico's sea floor, cataloging sediment types for oil and gas surveys, routine, procedural work nobody expected to make headlines. Ordinary oceanography assumes water is water, that if you drop a submersible into the sea, you get more sea, uniform salinity, uniform density, all the way to the bottom. Then the ROV's camera reached a depression in the seafloor and stopped, because there was a lake down there, inside the ocean, with its own shoreline, its own waves, and a mirror-smooth surface reflecting the vehicle's own lights back at it. NOAA researchers documented the phenomenon during multiple 2014 to 2016 Gulf of Mexico expeditions, finding brine pools with salinity roughly 5 times that of the surrounding seawater, dense enough to sit as a distinct, motionless body at the bottom of the ocean floor. Sometimes nicknamed by the crew the jacuzzi of despair for the mats of dead crabs found along its rim. Early hypotheses suggested these were simple salt seeps with no ecological significance. That collapsed once researchers found dense mussel beds and shrimp colonies thriving along the pool's edges, feeding on chemosynthetic bacteria in water that kills nearly anything that swims into it directly. Other scientists propose the pools were geologically temporary. Some have instead persisted for thousands of years, an ancient ocean, buried inside a younger one, still sitting exactly where it settled. Somewhere on this planet right now, there is a lake with a coastline that no human being has ever stood on, older than most of recorded civilization. And it is at the bottom of the same ocean your city's coastline touches. What is still settling at the bottom of the pools we haven't found yet? Whatever lives at the edge of that poison lake had to solve a problem stranger still. Number 8. The Xenophyophores. British naturalists studying deep-sea sediment in the early 1880s aboard the MMS Challenger expedition were mainly cataloging mud, temperature, and current data to chart the ocean floor for shipping and telegraph cable routes, unglamorous survey work. Cell biology in every textbook holds that a single cell has hard physical limits. That once an organism grows past a fraction of a millimeter, it needs specialized organs, blood, and a nervous system to function. because diffusion alone cannot service a body any larger. Buried in trench sediment, researchers kept finding strange star-shaped or spiral organic structures the size of a human hand, and for decades nobody was certain if they were looking at a fossil, a mineral formation, or something alive. Marine biologist Lisa Levin at Scripps Institution of Oceanography, working with colleague Andrew Godet, confirmed through later dives that these structures, Xenophyophores, are in fact single cells, some growing past 20 centimeters across, making them among the largest individual cells ever documented on Earth. Godet's team published further findings in Biology Letters in 2017 after surveys of the Clarion-Clipperton Zone in the eastern Pacific, describing unexpectedly dense Xenophyophore populations at depths exceeding 4,000 meters. Early explanations proposed they were simply slow-growing colonial organisms, not true single cells. Genetic sequencing ruled that out. Others suggested their giant size was possible only because deep trenches lack predators. That explanation weakened once Xenophyophore remains were found bearing what looked like feeding scars. A single cell, larger than your fist, has been quietly solving a problem that should be biologically impossible. At the bottom of an ocean, most of us will never see the floor of. If one cell can grow that large in the dark, what governs the ceiling on the ones we haven't measured yet? That single cell was mapping the trench floor before we ever named it. Number 7. The Osedax bone-eating worms. In 2002, marine biologists Robert Vryenhoek and Craig Smith were studying a routine whale carcass that had sunk to the Monterey Canyon floor, tracking how deep-sea scavengers strip a whale fall down to bone over a period of years, a well-established ecological pattern. Standard decomposition science says bone, once stripped of soft tissue, is essentially inert, too dense and mineral-locked for most organisms to extract nutrition from without teeth, acid glands, or a digestive tract built for it. Instead, the whale's bones were covered in what looked like a red carpet of thin, root-like worms, boring directly into solid bone with no visible mouth, stomach, or anus. Vryenhoek, along with Shauna Gofrady and Greg Rouse, formally described the species in a 2004 paper in Science, naming the genus Osedax, Latin for bone devourer, and finding that the worms have no digestive system at all, instead hosting symbiotic bacteria inside root-like tissue that dissolves bone directly using acid and enzymes. The failure parade here is short and strange. Scientists first assumed the worms were simply eating marrow through an undiscovered mouth structure. Dissection ruled that out entirely. No mouth exists. Others proposed the bacteria alone were responsible, and the worm was just a passive shell. That failed once researchers confirmed the worm actively secretes acid to open the bone before bacteria ever engage. Rouse's team then discovered something stranger still. Males of the species never grow past microscopic larval size, living their entire lives lodged inside the female's tissue. A permanent, dwarfed harem. Every whale that has ever died and sunk to the ocean floor across geologic time has likely fed this animal. Your own skeleton, given enough centuries at the bottom of the sea, would not be an exception. Whatever built that worm's chemistry had already been tested on something larger, 30 meters long. Number 6. The giant Siphonophores. In April of 2020, scientists aboard the research vessel Falkor, operated by the Schmidt Ocean Institute, were conducting a routine mapping survey of Ningaloo Canyon off the coast of Western Australia, cataloging seafloor topography for a broader biodiversity study, nothing designed to find a headline creature. Marine biology generally treats colonial organisms like Siphonophores as modest in scale, coiled structures a few meters long at most, distant relatives of jellyfish built from thousands of genetically identical, specialized clones working as one body. At approximately 630 meters down, the RUV's camera drifted along what the crew initially mistook for a geological feature, before realizing it was alive, and it kept extending and extending. Well past the edge of the frame, researchers aboard the expedition, in coordination with marine biologist Nerida Wilson, measured the organism, an Apolemia species, at an estimated 45 meters in outer ring diameter, among the longest animal specimens ever recorded on film, longer than a blue whale. The Schmidt Ocean Institute published footage and preliminary findings publicly in 2020, and the specimen remains one of the largest documented Siphonophores in scientific record. Early speculation proposed the organism was several individual Siphonophores tangled together by chance. Genetic and structural analysis of the feeding pattern made that unlikely. The outer ring behaved as a single coordinated hunting structure. Individual zooids working in concert like fingers on a hand no one owns. Others suggested the length was an illusion of camera perspective in murky water. Frame-by-frame measurement against the RUV's laser scaling grid confirmed the scale was real. A single animal, longer than the largest creature that has ever lived on this planet, was drifting through open water. And no one on the surface had any idea it existed until the footage was already rolling. How many bodies that size are moving through water we have simply never pointed a camera at? Below that drifting giant, the pressure itself starts rewriting the rules of chemistry. Number 5. The Piezophile bacteria. In the 1990s, microbiologist Douglas Bartlett at Scripps Institution of Oceanography was investigating how deep sea fish and bacteria tolerated crushing pressure at extreme depth, part of a broader effort to understand pressure adaptation in marine life for basic biological science, not headline-chasing research. Cell biology holds that extreme pressure. Over 1,000 atmospheres in the deepest trenches should denature proteins, collapse cell membranes, and halt metabolic function entirely. The same principle that would instantly crush human tissue and rupture organs on contact. Bartlett's lab instead isolated bacterial strains from Mariana Trench sediment, organisms later classified as Piezophiles, that were not merely surviving crushing pressure but actively required it, growing sluggishly or dying entirely if brought to the pressure of the surface. Bartlett's team, publishing findings across multiple papers through the 1990s and 2000s in journals including Applied and Environmental Microbiology, documented species like Moritella yayanosii and Schuonella benthica thriving at pressures exceeding 1,035 atmospheres, roughly the weight of 50 jumbo jets pressing down on a single square inch. Early explanations proposed thicker cell walls as the sole adaptation. That was only a partial answer. Later research revealed entirely restructured cell membranes using unusual fatty acid compositions that only function correctly under extreme compression. Other scientists proposed these organisms could be gradually pressure trained to survive at the surface. Most piezophiles simply cannot. Their proteins are so specifically evolved for compression that the shape denatures irreversibly once the pressure is removed. These are organisms whose biochemistry makes no sense anywhere else in the known universe except inside a very specific narrow band of crushing darkness. If your own body were lowered to that depth, it would not adapt. It would simply stop being a body. Down there, in that same crushing dark, something else has been making sound that no one can source. Number 4. The unidentified deep sea vocalizations. Throughout the 1990s, the United States Navy's SOSUS hydrophone network, originally built during the Cold War to track Soviet submarines, was repurposed by NOAA's Pacific Marine Environmental Laboratory for oceanographic and marine mammal research, a routine reassignment of military infrastructure to civilian science. Bioacoustic science generally assumes that any large, loud biological sound in the ocean can eventually be matched to a known species by comparing its frequency signature and pattern against a growing reference library of whale and fish calls. Starting in 1991, NOAA researchers recorded a sound they nicknamed upsweep, a rising, narrow band call detected across the Pacific Basin, repeating seasonally for years, that has never been definitively matched to any known animal, ship, or geological process. NOAA acoustician Christopher Fox and colleagues at the Pacific Marine Environmental Laboratory documented upsweep continuously from 1991 onward, noting its seasonal intensity peaks in spring and autumn, publishing spectrogram data through NOAA's public hydrophone archive without ever closing the case on its origin. Early explanations proposed a large whale species performing an unusually structured call. That explanation weakened because the sound's acoustic profile does not match any cataloged cetacean vocalization, including species known to produce low-frequency calls. Others proposed underwater volcanic or seismic activity near the presumed source region in the South Pacific. That remains only a partial explanation, since the sound's amplitude has measurably declined over the decades in a pattern inconsistent with typical geological activity. The source location has been narrowed, roughly to a region near the Pacific Antarctic Ridge, but the sound itself remains formally unidentified in NOAA's own records to this day. Every year, for at least 3 decades, something in the deep Pacific has been making a sound loud enough to cross an ocean, on a schedule, and not one scientist listening has been able to say with certainty what it is. Whatever made that sound would have needed an entire ecosystem no one expected to survive without sunlight. Number 3. The hydrothermal vent chemosynthetic ecosystems. In February of 1977, a team of geologists aboard the submersible Alvin, led by Jack Corliss of Oregon State University, descended to the Galapagos Rift to study underwater volcanic activity and plate tectonics, a mission with no biological objective whatsoever. Biology at the time operated on one settled foundational rule that essentially all life on Earth depends, directly or indirectly, on photosynthesis and sunlight, the base of every food chain ever documented. At roughly 2,500 meters down, in total darkness, Corliss's team found mineral chimneys venting scalding, chemical-rich water, and clustered around them, dense colonies of giant tube worms, clams, and crabs, thriving, with no sunlight reaching them at all. Corliss and his colleagues published the discovery in Science in 1979, describing an entirely novel ecosystem built on chemosynthesis, in which bacteria convert hydrogen sulfide from the vent water into usable energy, a biological process previously considered a marginal curiosity rather than the foundation of an entire food web. The failure parade began immediately. Some scientists proposed the animals were simply feeding on organic material drifting down from the surface, standard marine snow. Isotopy analysis of the tissue ruled that out. The carbon signatures matched the vent chemistry, not sunlight-derived carbon. Others argued the ecosystem must be a rare, isolated anomaly. Since 1977, comparable vent ecosystems have been found on ridges across every ocean basin on Earth, suggesting the model is not an exception. It may be one of life's 2 independent starting templates. The implication reaches further than the ocean floor. If life on Earth can build a complete, self-sustaining biosphere with zero sunlight. The requirement scientists once treated as universal for life anywhere in the universe was never actually true. If sunlight was never the rule, what else did we assume was required for something to be alive? Nothing on this list has resisted explanation longer than the sound recorded in 1997. Number 2. The bloop. In the summer of 1997, NOAA hydroacoustic researcher Christopher Fox and his team at the Pacific Marine Environmental Laboratory were using the same repurposed Cold War hydrophone array to monitor undersea volcanic and seismic activity across the eastern equatorial Pacific routine environmental monitoring work. Standard acoustic classification holds that any sound powerful enough to be detected by multiple hydrophones over 3,000 kilometers apart must originate from either a massive geological event or an extremely large, well-documented marine animal, with very little room in between. In August of 1997, sensors more than 4,800 kilometers apart simultaneously picked up an ultra-low-frequency sound, nicknamed the bloop, whose acoustic profile rose and fell in a pattern that early analysis flagged as resembling a biological call, far louder and different in shape from any known whale. Fox's team at NOAA's Pacific Marine Environmental Laboratory logged and publicly released the recording in the early 2000s, noting the sound's profile did not cleanly match existing marine mammal vocalization records in their database at the time, which briefly fueled speculation that something larger than a blue whale, the largest animal known to science, was responsible. The failure parade unfolded over nearly a decade. Cryptozoological explanations proposing an undiscovered giant animal were the first to be publicly ruled out, since no known biological structure could physically generate a call at that volume and depth. In 2005, NOAA scientist Robert Ziak and colleagues matched the bloop's acoustic signature to icequake events. The fracturing of Antarctic ice shelves, a conclusion largely accepted since. What lingers is smaller than the mystery itself. The original spectrogram still resembles biological calls closely enough that researchers cross-checked it against whale vocalizations before the ice explanation ever surfaced. The case is closed on paper. The shape of the sound has never stopped looking like something alive. Everything on this list together points toward the deepest point on the planet and what was found waiting there. Number 1. The Mariana Trench's Unclassified DNA. In the late 2010s, a joint team of Chinese researchers, including microbiologist Wei Zi Song and colleagues affiliated with Ocean University of China, were conducting a routine metagenomic survey of Mariana Trench sediment, cataloging microbial biodiversity for baseline environmental science, standard census work with no expectation of anything unprecedented. Genomic science generally assumes that once environmental DNA is sequenced from a sample, the vast majority of genetic material can be matched, at least at a broad level, against known branches of the tree of life, bacteria, archaea, or eukaryotes, even if the specific species is new. Instead, sediment pulled from Challenger Deep, nearly 11,000 meters down, the deepest point in any ocean on Earth, returned genetic sequences that would not cleanly align with any previously cataloged phylum, forming clusters of DNA that sat structurally apart from the rest of the tree entirely. Researchers publishing Hadal zone microbiome studies through the late 2010s and early 2020s, including work supported by Ocean University of China and international collaborators, documented that a significant proportion of microbial sequences recovered from Challenger Deep sediment could not be matched to any existing genomic database, suggesting the trench harbors lineages that have likely been isolated from the rest of Earth's biosphere for a very long time. The failure parade here is ongoing, not resolved. Some researchers proposed contamination or sequencing error as the simplest explanation. Repeated sampling across multiple expeditions returned the same unclassifiable clusters, ruling that out. Others proposed these were simply rare variants of known bacteria, not truly novel branches. Deeper genomic comparison found the divergence too structurally significant for that explanation to hold cleanly. What remains is genetic material, sitting at the bottom of the deepest trench on the planet that our own species has not yet found a place for on the only family tree we've ever built. Every strand of DNA in your own body traces back through that same tree, standing on a branch we have mapped. Something down there is standing on one we haven't. 10 discoveries, 10 teams of scientists who were not looking for any of this. Every one of these entries did not appear at the edges of biology, where uncertainty is expected. They appeared inside routine surveys, inside settled fields, inside the parts of science we assumed were finished. The ocean has not been hiding these things from us. We simply have not gone deep enough, often enough, with the right instruments pointed the right way, to notice they were already there. The trench is still down there tonight, unclassified, unresolved, indifferent to whether we ever finish mapping it. If you want to see more videos like this, click the video on screen now.